So our next speaker, as Dr. Bringhoff mentioned, is Professor Jeff Nivala. He also has been working on nanopore-based proteomics for a while now in his PhD. As alluded to in the last talk, they developed or utilized ClipX to try and sort of sequence full proteins to many advantages. And then today he's going to be presenting on his work, sort of expressing multiplex tags based on things that they learned during his PhD and after as well. Looking forward to your talk.
Hello there, everybody. Can you hear me?
Yes, we can.
Okay, fantastic. Can you see me? I'm just curious.
Also, yes.
Oh, okay, great, fantastic. Okay, how's everyone doing? Yeah, so thanks first off to Nikolai for the invitation and allowing the flexibility for me to present remotely here from Seattle. Wish I could have joined you all. So yeah, I'll be talking a bit about some different routes that we're exploring to go towards developing nanopore-based proteomics, starting from reading or sensing peptide barcodes all the way to some full-length proteins. So it's a tough act to follow with Henry's awesome work, but also a good setup to some of the questions that people were asking about, maybe some possible paths towards full-length protein analysis. Okay, so my group here is sort of broadly focused on molecular technology development using both synthetic biology tools and nanopore technology for reading and writing data in molecular form. So going from molecular recording systems, DNA data storage, to also next-gen proteomics methods, new ways to read proteins, and it all sort of is based, a large part into developing, using nanopore technology for new applications. And as Henry just gave a nice introduction to how these devices have been commercialized now for DNA and direct RNA sequencing, and what's really making them big game changers, not just for DNA sequencing, but sort of all sort of molecular sensing, because they are, just essentially generalizable molecular sensors, is that they're incredibly portable, inexpensive devices that can plug into your laptop, even your smartphone, right? So my group sort of thinks about what new molecular apps can we use these type of technologies for now, and looking, sort of very far off into the future as well. Okay, so of course today I will be focusing on some of the work we've done with single molecule proteomics. I just wanted to start off by mentioning, sort of drawing a parallel between, I think this is the fifth single cell proteomics meeting, dating back to 2018, and in 2017, around the same time, there was the first single molecule protein sequencing conference was organized and held in Delft in the Netherlands, organized by Kees Dekker and Truman Jew's lab there. And I think the third one, it's a biannual, so I think the third one is going to happen October of this year, so definitely folks should look into that. It'll be in Delft as well, for people to sort of see the latest in single molecule protein sequencing efforts, and it goes beyond nanopores for sure. So super interesting technology there. And I will say, just seeing the talks today, it seems like the single molecule, or sorry, the single cell folks have been kicking the single molecule people's butts, and so we have quite a bit of catching up to do. So some of the challenges associated with proteomics, and maybe in single cell proteomics specifically, could those potentially be solved with single molecule protein sequencing, sort of getting at the motivation here. So as was covered widely today, a major challenge in proteomics is that high dynamic range, and so you'd like to be able to have some ability to be quantitative over that high dynamic range. So single molecules are in a way inherently quantitative, because you're essentially counting molecules as you observe them, single molecules as you observe them. And then there's also tons of intraprotein heterogeneity, right? So isoforms, post-translational variants, these sorts of things. And just sort of showing you one example here, everyone's favorite protein P53 and all the different, this is just a very small subset, but there's many different proteoforms of this particular protein, and of many important proteins within the cell. And each of these different proteoforms can have very different biological activities within the cell. Okay, and then sort of going back to the ability to look at full length single molecules, a lot of these effects can be occur over long ranges and combinatorially. So the ability to read a molecule in its full length state and be able to correlate these effects, I think would be really important compared to traditional technologies such as mass spec that are relying on fragmentation, where you're losing this information. So really, your ideal protein analysis method, at least from my perspective, would give you that single molecule resolution, and it would keep proteins in a native full length state to avoid that fragmentation step. Okay, and if you look at the most popular existing proteomics technologies, they are not single molecule, and they are not giving you essentially full length reads as we might call them. So this is where nanopore comes in. Fortunately, I can skip over how nanopores work, nanopores 101, because Henry gave the great Spark notes there. But one key point that Henry also mentioned is sort of the breakthrough in nanopore-based DNA and RNA sequencing was the ability to sort of two combinations of things come together to make the secret sauce. It's motor-based control, using that enzyme to ratchet that DNA strand through the pore, and then having a pore with the right structural dimensions, such that you're sensitive to single nucleotides, single nucleotides. Okay, and so these are the same concepts that we think we need to apply towards single molecule protein sequencing with using nanopores. And so around the same time as this breakthrough that I was just talking about, Oxford Nanopore has commercialized now the first commercially available nanopore-based sequencers, and as Henry mentioned, some of the great things about nanopore sequencing: unlimited strand read length, single molecule, sensitive to modified bases, portable, and expensive. Okay, we'd love to have all these things for protein analysis. Most protein labs, and are using this, most nanopore labs are using this sort of electrophysiology like setup here, using a patch clamp amplifier, collecting data from a single nanopore at a time. And this is where I did essentially the bench, actually, where I did my my PhD work in Mark Agison's lab, to collect some of the data that I'll be talking about today. But essentially, once I graduated here, I said I'm never going back to one of these things again. It's incredibly technically challenging to set up one of these experiments, and quite painful and very low throughput in the amount of data you can collect. And so it's really not as scalable in this form, and it's also not usable by non-experts. And so instead, I got my lab. What I really wanted to do is make sure technologies that I develop can be more easily translated to other labs and industry and possibly into the clinic. So what we've done is really try to use off the shelf commercial devices as much as possible, particularly these what they call the MinION devices here, which have 512 parallel independent nanopore channels. And so we can pretty much pull these devices off the shelf, like I said, and then essentially hack them, not just for DNA and RNA sequencing, as most people who are using them are doing, but actually use them as high throughput nanopore sensors for new applications. And I'll talk to you about how we're using those for proteins. So just a bit about them. Like I said, it's a nanopore array, so hundreds of individually addressable nanopores are in there inside of a flow cell. Everything is in liquid phase in there, and you can basically pipette any sort of samples that you want into there and collect all the data in parallel from all these different nanopore sensors. It's also really nice. It's a very scalable technology, down to devices that have a few dozen pores that are disposable, all the way up to devices that are used right now currently for like high throughput genomics projects, where we have hundreds of thousands of pores you can collect data from. So lots of big breakthroughs have happened in genomics and transcriptomics. This is an outdated slide now. I could add much more much more here because you're getting sort of nanopore enabled breakthroughs in your ability to read DNA and RNA. So what about the proteins? So like I said, we sort of have been asking what proteomics can we get established using these commercially available nanopore platforms. So what we did, we started by just starting really simple. What do protein strands just look like in a nanopore sensor? After this, we started developing some immediate applications, particularly some multiplexable peptide barcoded reporter proteins. So I'll give a brief overview of how those work, and then I'll sort of go back and touch on the analysis pathways for enabling full-length protein analysis using unfold-based motors for full-length protein identification or sequencing. And then because this is a single-cell conference, I thought I just might throw out some wild speculation of how single-cell nanopore proteomics might work and what maybe some advantages that nanopore sensors have there. So challenges of detecting full-length native proteins with a nanopore sensor is compared to DNA and RNA is that proteins are non-uniformly charged. So you can't rely on just electrophoretic effects to drive translocation or targeting of your protein to your nanopore sensor. And they also are adopting large tertiary structures that aren't going to fit or very nicely thread into that pore. So how do we go about sort of detecting one of these native proteins with a nanopore sensor? And so this is the way we designed the problem. Designed around the problem is basically you can take your folded domain and you can attach a negatively charged tail to it. Okay, and this is in particular, this is at the C terminal. And so negatively charged tail, it's primarily made up of glycines, serines, and then aspartic and glutamates to give it that flexible negatively charged nature. And what essentially allows us to do then is proteins that are tagged with this at one end with this tag essentially can then be electrophoretically driven into the nanopore sensor under an applied voltage. It doesn't translocate for this particular domain, typically does not unfold under that force, right? You're imparting a electrophoretic force by pulling onto that tail, but that folded domain is sufficiently mechanically stable to be able to resist that, and essentially it will just immobilize that peptide strand within the pore. Okay, and what this system allows us to do then is essentially modify the peptide sequence of that tail, and then we can see how changing the sequence of amino acid changes the nanopore signal that we observe. So it's a really nice system to sort of interrogate different protein sequences. And so using this sort of model, the first thing that we did, we sort of wanted to come up with a model of how many amino acids actually fit within the protein nanopores that are on the MinION devices. In particular, they're using a variant of the CSGG nanopore. And what we found using a series of mutants here, essentially we took this sort of baseline glycine, serine, aspartic acid tail sequence, and we introduced a sliding window of tyrosines across a series of different mutants, and then see how the nanopore signal changed with each of these mutants. And as you can see, as we sort of slid this sliding window down along the peptide tail here, we're gradually occluding more and more current, and then gradually going back up and sort of returning to baseline. And what this allows us to do is basically come up with a model of, okay, how many amino acids, when we're threading it through the pore, are contributing to the ionic current, how many are actually fitting within the pore sensitive lumen, and what is their effect, what are their differential effects on the current level. And doing this, we saw about 17 amino acids fit inside this particular pore. And they all are sort of generate, depending on where the mutation is at along that peptide tail, sort of generates differential effects on different features of that ionic current signal. But essentially what that allows us to do is we can take some collection of these different signal features and basically cluster them and see that they can have very unique signals, at least from this set of different mutations. Which were mainly designed just to generate large deviations in signals such that we could map the pore in. So again, but that also means that there's 20 to 17 possible sequence combinations that you theoretically could have to resolve against if you're trying to do sort of de novo sequencing using this approach. Some other interesting things that we found using this sort of immobilized peptide sensing method is that the nanopore signal roughly correlates with the amino acid volume. And we can call this sort of the amino acid barcode, this region that is actually being sensed. And essentially bigger amino acids include more of the ionic currents in general. And so we also looked at some, sensitivity single residue modifications. And so this is one particular, we call the phospho, took a PKA kinase motif, we cloned it into this, this peptide barcode, mutated it to compare the traditional kinase motif to a phosphomimetic mutant by making a serine to glutamate modification and seeing that it resulted in a large deviation in current. So sensitivity, at least in some contexts, down to single residue modifications, which then inspired us to actually see if we could detect real post-translational modifications. So in this case, we were looking at activity of the CK2 kinase, where we engineered in a CK2 kinase motif into that barcode region, incubated it with the kinase, and then measured its nanopore signal over time. And we could essentially track the phosphorylation status of that barcode while it was being incubated with CK2 over time, because it resulted in this large shift in the nanopore, in that barcode's nanopore signal. So of course we were sort of climbing this tree towards single molecule protein sequencing, but along the way, we sort of wondering what can we pick any low-hanging fruits along the way, and so as we sort of we're looking at into these different barcode sequences by suspending them onto this folded domain, we realized that they could be useful actually as peptide barcoded reporter proteins. And so we recently published some work here where we actually showed that we could developed a new class of reporters that we call nanopore addressable protein tags engineered as reporters, or nanoporters, where essentially they are more multiplexable forms of fluorescent proteins. You of course are not going to get your spatial readout as you would with microscopy, but for many applications, people are using fluorescent proteins in genetically encoded systems, using a fluorometer to measure protein expression levels. It's limited in multiplexability because of spectral overlaps between fluorescent proteins, and that's a problem for small molecule fluorophores as well. And so we essentially can use the peptide barcoded technique, where you can essentially generate orthogonal reporter proteins by using a different barcode within this protein construct that I'm showing you here. And essentially, we've engineered these such that when they're expressed, they're secreted outside of the cell. And so really, it's really easy sample prep. Or essentially, you can just take a sample of the media, directly load it into a MinION flow cell, and be able to detect these different barcoded proteins as they're expressed. And the way, because we're not doing sequencing, the way that we actually do classification is pretty simple. We're just using supervised machine learning here, using the same sorts of classifying models that people use for computer vision. Really, just we really treat the signal like an image, and we're able to get 90 plus classification accuracies at the single molecule level, 20 plus in 20 plus way multiplexing. Like I said, direct loading of unprocessed we can do them in bacterial. We can also do these in human cells. I'm not showing you the data here, but essentially they're very modular, in that you can put them into different model systems for multiplexed gene expression tracking. Just results from a sort of 9-way multiplexing in E. coli. And let's see, we can be quantitative, because essentially the concentration of these different barcodes in solution is going to, the capture rate of these molecules, basically the rate at which you read them is dependent upon the concentration of them in solution, right? So you can use a standard curve to do some level of relative quantification across these different barcodes. And really for these experiments, less than 10 minutes of runtime is sufficient for accurate determination of nanoporter capture rates across around 6 orders of magnitude of concentration. Down in these, without doing any special tricks, down to the what is this? Down to like a nanogram range. But we think it'd be easier with more tricks. We can take that down by orders of magnitude. So really looking at from a reporter aspect, traditional fluorescent reporters are sort of, kind of like a lighthouse, a lot of spatial information, but limited total information content, whereas nanoporters, you sort of, not necessarily any spatial information, but high information total content, high total information content, because you can pack many of these barcodes into a single system potentially. So now this, various applications we're exploring for these. So how about towards sequencing, right? Analysis of full-length native proteins. So we need a motor to pull proteins through the pore, full-length proteins. So this is ubiquitous process in nature. Is your cells are importing and exporting full-length proteins in and out of tiny holes. And so one of these is this ClipXP complex, where ClipX is this unfoldase. It targets proteins for degradation, grabs onto them, mechanically unfolds them, and threads them into the ClipP protease for degradation. And the way that we're able to couple these for full-length analysis of native proteins is essentially we can take this negatively charged flexible tag. We can also, at the end of it, append an SSRA tag, and that's what ClipX recognizes. We can add our tag protein analytes to the cis side of the solution, but we captured into the nanopore, and then an unfoldase motor that we have present in the trans solution can then bind this tag and then catalyze unfolding and translocation of that domain through the pore. So this is what raw data looks like for a single domain protein. This is a double domain protein. Here's a 4-domain model protein that we engineered. And what you can see, very complex nanopore signal here, but essentially every domain has a unique pre-unfolding state and then a unique unfolding and translocation state. And this particular protein was over 700 amino acids in length. And so this is cool video sort of showing you what this sort of looks like. We won't need to watch the whole thing here, but essentially your tagged protein captured into the pore. There's your unfoldase motor. It binds it. It's going to translocate up that tail until it encounters the pore, and then it's not going to be able to move any further until it's able to start unfolding and processing these domains. And you're getting not just sort of sequence information as the different sequences move through the pore, you're also getting some structural information because it takes a variable amount of time for these domains to unfold depending upon the mechanical stability. So we can pick up different things such as unfolding intermediates, proteolytic cleavage events, these sorts of things, and resolve sequence dependent features, stability, as well as relative size of different domains. So this is sort of at the domain level of protein translocation. We can sort of feed all these signals, again, use supervised machine learning and come up with some very accurate single molecule classifications for detecting these particular proteins. We're also moving towards very similar to what the data that Henry just presented. So the previous stuff in the past slide, that was actually from my PhD work 10 years ago. Now we've picked it up in the last few years again, now that I've started my group, and we're pushing more towards single amino acid resolution. And this is just some data that we have. Very similar substrates to what Henry was showing you, but actually using an unfoldase motor to pull longer strands through that pore. So, stay tuned for some of this work. Just like Henry mentioned, sample tagging, minimal sample prep for this method. We just need to attach our tags to one end of the protein, and we can get this chemistry working pretty good. So sort of going back to the ideal protein single cell analysis analyzer here, I promise I pontificate how single cell, how this might work with single cell. And so a big challenge from my perspective in single cell sensitivity is really it's a delivery problem. And so you really want the minimum spatio-temporal distance between your sample and your sensor. And so this is where I think nanopores are quite unique in that your sensing is actually occurring in a very biocompatible conditions, right? We're using enzymes, things like this to process these molecules through the pore. So you could imagine using a device like this. This is a 96-well nanopore device in development by Oxford Nanopore Technologies. You could use existing single cell sorting technologies, sort single cells into each individual well here. And each well actually has its own nanopore array associated with it. And you could imagine doing lysis and sample prep end to end within that single well, also your sensors are at, right? So once you have that cell in there, it never leaves until you get the proteomics data out of it. Okay, and then more wild speculation. sort of what would throughput look like, assuming your nanopore motor processes proteins 100 amino acids per second, assuming average length of 300 amino acids per protein, and you had 100 pores per well, it would take you about an hour to analyze 100,000 single protein molecules from 96 single cells. Again, wild speculation, but that's you know, something that I could see possible in the future. And then also just nanopores, because we're using them for DNA, RNA, and now hopefully soon proteins, sort of this one technology where you could imagine getting genomics, transcriptomics, and proteomics data all from the same cell with single molecule resolution, epigenetic modifications, PTMs, all that sort of stuff. Who knows when that will happen, but that's the dream, right? And so with that, I thank my great students here, Katie, Nick, Karen, Erilyn, really leading on the nanoporter work, and then Keizuke Matone for doing the Unfold Days work. And thank you all for listening.
Happy to take any questions from the audience. Yep.
Thank you. Amazing talk. Do you think that this technology can be used for cross-linked proteins?
I do. I mean, I think you know, reducing the cross links using traditional, you know, reducing agents would be the simplest way to do that. There's no reason why that would be incompatible with the nanopore. Of course, you might lose some of the structural information there. There's no reason why ClipX can process disulfide linked beta strands, for instance. It does have a flexible sort of ability to process things that are irregularly sized, but as far as threading through the protein, threading through a protein pore with that disulfide linkage intact would be difficult.
Thank you. My question was, what can you tell about, or what details can you mine out of the unfolding process? And could you use, you know, basically your existing technology to monitor small molecule protein engagement, the stabilization to the tertiary structure that might happen?
Yep, for sure. So I definitely think that is possible. So in work that I've done with the Unfold Days, we definitely have looked at characterized unfolding intermediates. So we took many variants of GFP that have been circular permutated, these sorts of things, and being able to characterize that they unfold in different ways. And then as far as small molecule stabilization of protein domains, I do think it would be possible using the Unfold Days. But actually other people have shown that, for instance, have they shown it with nanopore? I think people have shown small molecule based stabilization or destabilization of folded domains just by pulling on them, sort of in a similar technique that I'm doing, but just relying on electrophoretic force. But yeah, go back to your point. Definitely, I think it's possible to use this technique to do that.
You could imagine an array of, for example, all known kinases physically tethered to a consumable, and then use it as a screen for small molecules.
Yeah, for sure. No, kinase I think is a super ripe. This technology would be great for screening kinases, and yeah, we should talk. I think that's a great idea there.